Good afternoon. Thank you for joining the Native Voices in STEM today. We really appreciate your time in this community and really excited to start this speaker series today. It's our final uh, talk for the fall 2026 semester. So yeah, it's Shea Carletta Chief Inishye. Good afternoon. My name is Carletta Chief, and I'm the director of the Indigenous Resilience Center and professor in environmental science as well as PI of the Native Fuse Alliance for the University of Arizona. Before we begin, I want to acknowledge that the University of Arizona is on the land and territories of Indigenous peoples. Today, Arizona is home to 22 federally recognized tribes, with Tucson being home to the Atop and the Yaqui. Committed to diversity and inclusion, the university strives to build sustainable relationships with sovereign Native nations and Indigenous communities through educational offerings, partnership, and community services. The Native Voices in STEM is a seminar series that has started many years ago. This series is co-sponsored by the Indigenous Resilience Center, the Native Fuse Alliance, the Sloan Indigenous Graduate Partnership, the Water Resources Research Center, and the Office of Societal Impact. This seminar series highlights scientists, engineers, educators, traditional knowledge holders, leaders, and social activists who are working with Indigenous communities. The seminar series aims to provide students with the opportunity to network and learn about the research and careers of leaders in these fields. Each session includes a seminar presentation as well as a Q&A at the end. All attendees are encouraged to engage with the speakers, ask questions, and participate in the discussion. After the seminar series, we invite students to stay with us for a one-on-one mentoring session with Dr. Cherie De Vore and engage in a mentoring topic with you. I'd like to now introduce our speaker today, Dr. Cherie De Vore. I'm so proud to have her here at the U of A and a faculty member in the Indigenous Resilience Center. Dr. Cherie De Vore is Diné. She comes from the beautiful high desert community of Crownpoint, New Mexico, on the eastern Diné Navajo Nation. She received her Ph.D. in civil environmental engineering at the University of New Mexico under the mentorship of Professor Jose Cerrato. She was awarded the National Science Foundation Earth Science Postdoctoral Fellowship to conduct research at Stanford University from 2020 to 2023. Dr. De Vore now leads the NVIS Biogeochemistry Environmental Engineering Laboratory Group as a new assistant professor in the Department of Chemical and Environmental Engineering at the University of Arizona. Her current work investigates biogeochemical mechanisms that affect mobilization of inorganic contaminants and microbial community changes for the purpose of engineering bioremediation efforts in rural and underserved community. Dr. De Vore is an associate faculty member of IRES and a co-PI on the Native Fuse Alliance, working to address food, energy, and water solutions in Indigenous communities. Dr. De Vore serves as the vice president of the University of Arizona Native Faculty Group. She is also the co-advisor for the U of A ACES chapter. Outside her research, Dr. De Vore enjoys spending time with her family, drinking coffee, growing food, engaging archery and marksmanship, hunting, martial arts, playing, collecting soul records, and participating in the ceremonies. It is my great honor to now give this platform over to Dr. De Vore. Thank you.
Can I ask if you can see my whole presenter window?
Yes, I can.
Okay, perfect. Thank you so much, Dr. T, for the lovely introduction and for the time and space to share the wonderful work that my students are leading here as I just finished my second year as assistant professor. I'd like to quickly acknowledge protocol in a formal introduction and greet all of you in the Zoom space today. So ja, at Cherie De Vore, Diné, Adon and Schlinigi, Yad Ashche and Schlin, Zelt Aki Ani Bashichin, Shiche Shihen, Danle, Ado Tratne Sahne Dashanele, the Kut Sanadon Schlin. So I'm Cherie De Vore, and I wanted to start with asking and engaging my audience what you think of when you hear the word abundance. So the title of my talk is Engineering from Abundance. In the chat, give me like a couple of words for what that means for you. Let's see. Glass full, glass half full mentality, enough of something, gratitude, plenty, plethora of things, sustainable, more than you need, restoration of local knowledge, endless, positive outlook, sustainability, a gift, overflow, rich in the things that keep you going, a lot of corn stored, presence. Okay, all of these resonate for me, and I have worked a lot on what it means to be an ancestor, a future ancestor, a relative, a community member, and a lot of the teachings that I've received from my family and community show up in the work that I do in engineering. And abundance for me starts with the way you think. It's a mindset, right? The way you navigate. And it's not, I challenge us to think it's not from a capitalistic perspective, tangible things or money, right? So I want to start in that way because something I've been challenged to think about in this work is the privilege, the incredible privilege it is to be a researcher. And positionality matters in research, and engineering research specifically, because engineers aren't just designing systems, we are designing impacts. Every choice we make, what problem to solve, whose data to trust, what risks are considered acceptable, and even what success looks like, comes from the researcher's standpoint. So naming your positionality doesn't weaken your objectivity, it clarifies your lens and exposes assumptions that you might have and strengthens the ethical grounding of your work. Engineering solutions also don't exist in a vacuum. I show up as the mentor and teacher that I am because of the family values, the core values that I carry. And I always like to acknowledge my mentors. So back home, those are cultural mentors, plant people. When I introduced myself, I told you I was born for red streak or red bottom people, and they are known to be healers and practitioners using plants. And then I'm born for Atop the Mountain Towering House, which makes reference to an actual living, breathing repository of knowledge and love on the high desert in New Mexico. And that clan are known to be practitioners, healers, chanters. I also come from land and seed stewards and lots of abundant ancestral knowledge. So more about my academic training. I received my PhD in civil and environmental engineering under the mentorship of Professor Jose Cerrato, Maddie Ali, Bruce Thompson, Carrie Howe, Lucia Rodriguez Fiere, and Eliane El Hayek. And then I did a few years of postdoc at Stanford University under the mentorship of Kabir Pei, who's a microbial ecologist, and Dr. Scott Fendorf, who's a soil biogeochemist. Spent a little bit of time at SLAC National Accelerator Laboratory, and then during that time was invited to take a position here at the U of A in the Department of Chemical and Environmental Engineering. So back to the solutions and importance of positionality is it helps us to see and highlight who benefits from our work, from the engineering work, who bears the burden of this work and the recovery necessary, and then understand how our background and influences can influence what we consider good or efficient. And this is important and so critical because most engineering frameworks have Western extractive efficiency-centered assumptions built in. So this allows us to naturally question, what does optimization mean? What forms of knowledge are being centered or excluded? And what are the long-term impacts, ecologically, community, and otherwise? So my journey is shaped by me being a first-generation matriarch from this high desert community. Bridging these knowledge systems requires humility, requires reciprocity and care, because I don't see my role as translating Indigenous knowledge into science. My lived experience also informs my research, my teaching, how I show up as a community member, and drawing inspiration from the tools and technologies in my own community and in your communities too, alongside story, which are meant to uplift. And that is the goal here. Having navigated institutions not built for people like me, I carry an awareness of that power of access and the importance of representation in engineering. So a few pictures here include on the top center is in the Nishtiji steam pit, which our people have used for generations to create accessible, storable food. So food security. The bottom right is the mathematics and the engineering involved in building tache, which is our sweat lodges. And then our hogan at the bottom center is a lot of engineering in motion, as I like to call it. So having this awareness and centering in those values of celebration through creation helps us to remain answerable, not just to your discipline, but to your community, to land, to future generations, and the relationships your research then touches. Your identity is not a bias. My identity is not a bias. It's a knowledge system. So values like restoration and abundance shift engineering away from extraction towards regeneration. And restoration guides engineers to ask, how can my system repair what has been harmed? And how does this design return something to the land and not just take? So with that in mind, I've worked for 10, 12 years as an environmental engineer investigating issues related to a lot of really traumatic history for our people around the land, around contamination and metals mobilization, and then more recently, understanding what role destructive fires have on ecosystem recovery. So here's a map of, in the green hashed marks, federal and tribal lands in the Western US, and then all of those colorful marks and dots there are hard rock abandoned mines. And for those that are located near the green hashed marks, there's something like 160,000 plus abandoned hard rock mines located within 10 kilometers of federal and tribal lands. So you can imagine that represents a unique route of exposure for our people. Thus, the research that I've done related to mining legacy and extraction is linked to some traumatic history and loss and destruction. And so subsequently, stories told about us and our people are told from that deficit and disparity lens. But where there is destruction, there can be restoration. And I lean into my people's origin narratives around Nayet Nezgane and Tobajis Chine, Monster Slayer, and Child Born of Water in my growth as a scientist. And this is critical for me as I've intentionally changed and reworked this overview to capture questions of what does recovery and restoration look like? And themes have emerged from this work when you consider your lived experiences and as researchers working with community and coming from a community, we have to be really careful about how our data is managed, how our stories are being told, and then how that can be applied and adapted to cultural needs in the dynamically changing environmental sciences and engineering. And it's important to note that as scientists and engineers, we have to work within what is called protocol, right? EPA has them, several government and research and state institutions have them, but they focus more on analytical precision and compliance from the field to the laboratory scale. But Indigenous knowledges and governance around our data are anchored at local and relational scales, which are grounded in specific spaces and places and waters and communities that consider not just us as humans, but non-human relationships as well. So as you can imagine, Indigenous data sovereignty is paramount for leading and grounding this work. And how do we do that? This has been over a decade of figuring out how to operationalize and even articulate what this would look like in science and engineering. And I've been very lucky with the mentorships, with the mentorship I've had, excuse me, and with the opportunity to mentor, to center relationships as the foundation and the seed of mentoring manifesting in different roles that we all play in our communities, because each of us does play a role. Within the landscape of environmental science and engineering, that's been for me from an undergrad to a postdoc to assistant professor. And then I'm being mentored by a full professor, and I have undergraduates all the way to PhD level students. All to say that data isn't neutral, it carries stories, context, and a lot of responsibility, but we can mirror this relationality and relationships at the foundation through mentorship, because then they mirror these principles. We have a manuscript coming out, and this is led by my undergraduate student from Hickory Apache. Her name is Jiraiya Collado, so look out for her in the future. I've often questioned what systems we are a part of for coming up with engineering solutions. And my family and my people reiterate to me that we've always been in relation to the land, and we've always been using the best available technology at those specific times. We've been in relation to the land, using sophisticated technology and philosophy in a very deliberate response to all of the changing social and environmental circumstances. I use that as a way to ground us in some work led by a student that just graduated with her master's, Nieves Vasquez, on this framework that we've come up with to articulate across scale, because as I mentioned, scale is really important, but also what you would consider, I'm going to quote Dr. Cajete's mechanical knowledge or tangible knowledge, which is what we deal with here in the science and engineering space. But let's also think about what is not seen by the naked eye, what is intangible or even metaphysical, if you want to call it that. So at the micro, what we can't see, but we can do, we can experience are things like microbes. We need a microscope to visualize these relatives, minerals, and even energy. At the macro scale, those can be things like research frameworks, what I'm talking about in this moment, philosophical protocols, and then even distinguishing the different ontological and epistemological knowings and ways of knowings, to what we can touch, feel, articulate, describe, in this research space. And for us, that's been the use of things like light microscopes and spectroscopy tools and biosafety hoods and the fungi that we work with for more community scale outputs, right? Like storytelling, zines, art, music. My group was just on KXCI for our first half of a recording that will be shared with the Tucson community on public radio. So that is one dream coming to fruition. And then in between all of this, relationality is the bridge for science communication, as well as what I've previously described as our values being assets and not biases. So the values of kinship and reverence, responsibility, respect, and reciprocity. In between all of these, of course, are things like modeling, data sovereignty, and knowledge systems. This work is being led by Nieves Vasquez, and look out for her manuscript on this soon. So this work has been many years in the making, coming into fruition into our research group, the Niche Environmental Engineering Biogeochemistry Research Group. And our focus areas are environmental biogeochemistry, including working with plants, soil, water, microbes, to address not just inorganic contaminants, which has been my focus, but more emerging contaminants as well, like PFAS and microplastics, while honoring and respecting Indigenous and traditional knowledge systems. I would love to talk about every single student and their work, but I'm only going to have time to highlight maybe one or two. So Ryan Cotter, who's an undergraduate, is leading work on what role desert mushroom podaxis can play on the uptake and accumulation of copper mine waste here in the state of Arizona. There's some work being wrapped up around what role microbes, fungi, and bacteria in soil ecosystems can play on recovery. Savia Gutierrez is leading work with amaranth and sunflower on the uptake and accumulation of copper mine waste from arid desert landscapes. And then we have Jeffrey and friends at Stanford leading work on what role P. microspora, Pistillaris microspora, which is a fungus, a filamentous fungi, what role it could play on plastic degradation. Macy Wynn is leading work on this hydrogel called calcium alginate, encapsulated with fungi, on the removal and immobilization of contaminants such as copper and arsenic. And then I'll spend the rest of the time with you talking about this biopolymer project with fungi for mixed metal contaminants. I gotta say, I'm really proud of these two students, all of the students. This work, almost ready for submission into a publication, is led by undergrads. These are not graduate students or PhD students. Jeffrey is a senior in chemical engineering, and Gwen Flores is Pascoyaki, and she's a junior in material science and engineering. And this work was one of the first babies of our research group on understanding alterations of the fungal biomass, surface chemistry alterations, with and without phosphate in mixed metal contaminated systems. And we're using inspiration from the landscape, of its regenerative capacity, of its resilience, for fungi specifically, and the very, very long-term use for bioremediation applications. And it's inspired by lots and lots of years of community work in areas that have been contaminated and impacted by mine waste. And what I have here is a figure at the field scale from on the top left at the mine waste source. This is Homestake Mine near the Black Hills, South Dakota, which operated from the mid 1800s all the way to the late 1970s. And the community of Cheyenne River, Lakota is still experiencing contamination to this day. So 150 miles upstream of the Cheyenne River community is this mine. It's been transported, suspended, buried, resuspended, and then accumulated in this soil profile here on tribal land. And you can kind of see this green riverbank vegetation, and that's all 30 plus medicinal plants and edible plants used by the community. So we had some questions posed on, well, one, is this being mobilized in our waterways? Is it being uptaken by fish? And is it being uptaken into our plant systems? Plants that we eat, like chokecherry, for example. And I would say the vast majority of this work goes unpublished, and it sits with the communities that have these questions, and where I'm able to draw some mechanistic insight to think about long-term remediation is on understanding some mechanisms, both abiotic and biotic. And if you're interested, you can read this paper about what role phosphate, which is a nutrient amendment for this area that's 90% dependent on agriculture for their economy, and bicarbonate, because this river is incredibly well buffered. So bicarbonate and phosphate on the role of ion displacement of arsenic into the Cheyenne River watershed. Desorption and mineral dissolution are other mechanisms to consider. But when we think about what role microbes like bacteria and arsenic can have, this paper looks at the way that microbes can respire. In other words, the way you and I breathe in one form of oxygen and breathe out another. These microbes can breathe in, if you will, one form of metal and breathe out another, altering its speciation, altering its mobility, altering its solubility, and they don't have to happen in isolation. This paper talks about what role iron and manganese can have on mobilization. And then finally, detoxification mechanisms of fungi specifically isolated from these sites. And with that in mind, some of the critical foundational data that's inspiring work led by Jeff and Gwen is this work with collaborators at UNM on what role endophytes or filamentous fungi can have on not just tolerance, but transport and immobilization. And this work was done, geez, over 10 years ago at this point by Jen Rutgers, who is a biologist at UNM, who partnered with us to isolate from a very large longitudinal study, as you can see here, what endophytes are co-occurring with what plants. And why this is key is that filamentous fungi co-occur with 70–80% of terrestrial plants, and mostly a symbiotic relationship, but sometimes in a parasitic relationship. So those associations can do a lot for ecosystems by increasing and uptaking more organic carbon, helping to scavenge nutrients like phosphorus and nitrogen, improving microbial biomass, enhancing soil enzymes, increasing root and shoot length. And endophytes, specifically with this project, are those that are inoculated at the seedling stage and then grow upwards into the aerial parts of the plant. Here's an example of little blue stem for this project grown with Cataphora species, which is a metal-tolerant fungi from one of these mine waste sites. And you can see just the extensive root system of this particular plant. And here's a cleared stain light microscope image of the fungi colonized inside of the plant. We can also visually see what role endophytes play on overall plant health. The picture of the plant on the left had no endophytes, was grown hydroponically, and a lot of them were very poor looking and didn't survive. Those on the right, though, were greener, had longer roots, and overall increased biomass. We expose those plants to different concentrations of arsenic inspired by the contaminated system there in Cheyenne River, and found that within the first 12 hours of exposure, most of that arsenic was removed. So where did it go? We then measured within the plant, with and without fungi. In the case that there was no fungi, most of that accumulated onto the roots, and those with fungi were able to uptake some of it into the stem. And the question is, why was that the case? Here's a scanning electron microscope image of, on the top, the roots of those plants without fungi. And we can see mineral phases with arsenic that are indicative of calcium phosphorus minerals. And then cataphora inoculated plants on the bottom, those mineral phases are phosphorus, magnesium, and calcium minerals. Now, it has to make it into the plant somehow. There are several pathways to do this, including moving all across the cortex here, past the pericycle, and then finally up the xylem, which is where nutrients and water make it to the plant for its survivability. However, because we inoculated with fungi, those hyphae become extensions of the root hairs, which is what you see here. Both the plant and the fungi have phosphate transporters. And because phosphate and arsenate are chemical analogs of one another, this is another mechanism or pathway into the plant. So if you zoom into these red circles here, which are our fungi inside of the plant, we confirmed with transmission electron microscopy that these fungi were indeed colonized inside of the plant, and that they could be one transport mechanism for metals. Now, here's a scanning electron microscope image at 100 micron scale, and you can see these little hair-like structures are extensions of the root. These are what the fungi look like. And if we were to zoom in just a little bit more, you can see. And then these are called conidia. These are reproductive parts of the fungi. So all of this to say, if this will advance, you look at what role fungi can play on helping with tolerance. This led us to even more questions than we began with, which is the inspiration for the work that my students are now leading. And those plants with fungi, they could tolerate arsenic. They even uptook arsenic a little bit, and we were able to confirm that with this osmium stain, which is attractive of organic material such as fungi. And that was the only place we were able to detect arsenic. In those plants without arsenic, you can see, without fungi, you can see the plants are really, really small relative to those with fungi, and then that these calcium, phosphorus, arsenic phases were sticking onto or absorbing, if you will, arsenic. So the heterogeneous distribution of arsenic throughout the plants with higher biomass enhanced their tolerance. We still have questions, though, about uptake and mechanisms for transport. Where we're at now is navigating between those years of work at the field and community scale and zooming into using these engineering, material science, biology tools at the laboratory and bench scale. And how we're able to do that, as I've nicely walked you through, is looking at surface water in areas impacted by mining and understanding chemical reactivity. Looking at soil, bacteria, and metals together. Surface water characteristics, metal characteristics with plants in hydroponic setting. Towards zooming in at the molecular scale for inside of the plant metals with fungi and metal solution for measuring absorption processes, which is a super hyper focused mechanistic focus, with the long-term goal many years from now for using these materials within an ecosystem context for bioremediation. Now back to work led by Jeff and um, why is her name not coming to me? Jeff and Gwen. Their specific measurable objective um, competes with metabolism dependent and non-metabolism dependent mechanisms for environmental systems. And there's a lot of overlap there as well. So there's one system led by Ignacio Garcia, who's looking at chemical reactivity specifically, no fungi or any living organism involved. Macy Wen is looking at inactivated fungi, so non-living fungi that have surface chemistry, surface charge that could participate in any one of these precipitation and physical adsorption reactions. And then those that are enzymatic and metabolic pathways for uptake, for volatilization, and for subsequent transformation of their toxicity. This is all in the very long term to design passive or active treatment unit ops using engineering tools. Multiple unit processes are needed with both biotic and non-biotic systems. This is necessary. If you were to look at a water or a wastewater treatment plant, it's based on a lot of this systems thinking. So their specific research objective for using fungi as an adsorbent is to integrate multiple tools like modeling and math, spectroscopy, microscopy tools, and characterize on the surface of that fungi after it's reacted with different metal contaminants, surface charge, surface chemistry, functional group alterations at these two pH conditions that were characteristic of those mine waste sites that were measured over these 10 years, right, under surface oxidizing conditions. And working with microbes is not trivial. It takes a lot of time and nurturing to grow these organisms. They're eukaryotes like you and I. They're more related to us than they are bacteria. To get them to grow non-contaminated, isolated, give them just enough food, wash them, not disrupt them, homogenize them, and then expose them to, as it says here, 2 parts per million uranium and 2 parts per million arsenic. And of course, we have adequate controls. Over a period of time, these students sampled what was in these flasks, and you can make a number of really cool models and isotherms based on understanding the mass balance within those batch reactors, right? What's removed and what stays in solution. Some cool analytical tools that these students have been very nicely trained on to answer some of these mechanistic questions include spectroscopy. So Fourier transform infrared spectroscopy, or FTIR, as I will refer to it going forward. Energy dispersive X-ray spectroscopy, which is often coupled with either transmission or scanning electron microscopy, light microscopy, aqueous chemistry tools like ICP-MS, ion chromatography, and a zetasizer, as well as culturing techniques. I start my students with understanding and knowing the literature and also doing some modeling to give us some intuition about what do we even expect in these systems, given the chemistry, given their solubility, given their speciation. And we found upon several iterations of this model that a lot of calcium-based minerals were being formed either with uranium or arsenic alone or them together. And then the most commonly predicted precipitates formed were calcium phosphate precipitates, as you saw in several slides ago in that hydroponic paper where the arsenic is stuck to or adsorbed to that root surface, right? We're seeing the same in this system here. And in those phases where arsenic and uranium and phosphate were together, those are highlighted in bold here, uramphite being one of them, and autunite species others. We also were visually able to see, using microscopy, the formation of these solids, these minerals, onto which subsequently arsenic and uranium can adsorb. So what's circled here in the red are crystals or minerals on the hyphae structure. So you look at these lines, and each of these panes, these are both Cataphora and Fusarium mineral-tolerant filamentous fungi. These squares are literally like salt crystals. And we wanted to figure out what exactly they were based on this modeling here, right? That gave us intuition about mineral formation, actually verifying that mineral formation, and then using SEM with energy dispersive X-ray spectroscopy to understand what phases these minerals were. And we indeed confirmed with this tool, as well as morphology characterization, that calcium phosphates appear as the predominant minerals at pH 4. And there is also natural, we think, potential phosphate solubilization by Tularamyces species, which is very indicative of what we've been reading about Tularamyces, a metal-tolerant filamentous fungi. We also use another tool, FTIR, to look at functional group alterations to then confirm what we're seeing both with light microscopy and EDX. And spectra changes before and after exposure to arsenic and uranium together with and without phosphate show bending and alterations of amides, phosphate groups, beta-glucan groups. Beta-glucans is especially important because that's the building block of chitin, which is the building block of the skeletal structure of fungi. So these changes indicate functional group involvement and alterations in metal adsorption. So big picture, plain language, these alterations are showing that indeed metal adsorption is happening onto our fungi and is altering the chemistry of that fungi. We also saw potential uranium-oxygen bonding, which says to us that uranium is altering the surface of the fungi, so adsorbing onto the fungi. We also detected hydroxyapatite formation, which is a calcium phosphate mineral, as I've been saying in this story a few times. Hydroxyapatite loves arsenic. They're like buddies in systems, and it's used in water treatment systems as a adsorbent and coagulant in most water unit ops in engineering. So the fact that we're able to see this with our fungi is very promising. So big picture, what are our data in this particular chapter telling us is that calcium phosphate mineral formation on these fungal surfaces were confirmed by multiple methods, right? By modeling, by spectroscopy, by microscopy, and by FTIR. These are so important and critical for our mechanistic understanding on mobilization because these mineral precipitates interacting with the fungal biomass can be the removal mechanism for these mixed metals. Where we're headed in the long term is to draw inspiration from the land, from these microbial relatives at the micro scale to understand on the left here. What I have for you is a photo zoomed in of what chitin and proteins look like on the cell wall membrane. And here's an enzyme or protein, I'm sorry, illustrating a metal ligand that could potentially be uptaken into the fungal biomass. There's a lot of mechanisms that we have yet to explore and are currently exploring on uptake. But what we've seen so far is that metal functional group surface chemistry alterations can drive removal. And we're going to, in the next year, use that knowledge to use electrospun nanofibers mimicking chitin in these materials to make our own biopolymer. And why this is attractive is these nanofibers they're low cost. They have very well characterized one-dimensional orientation, high surface area, high porosity, and they're also biodegradable and non-toxic and biocompatible, and they have a really unique structural arrangement. We have students right now in the lab analyzing our aqueous chemistry. So I haven't showed you anything yet about removal from solution, but we've got a lot of evidence here that arsenic and uranium are interacting with and are being retained and mobilized by our fungi. Oops. I want to acknowledge my lovely research group, all of the students here who work really hard and make the community a beautiful, vibrant space and such a pleasure to work with. Each of them has so many unique strengths and assets that they bring to the space. I also want to acknowledge the Indigenous Resilience Center, with whom I am an associate faculty member. And then a lot of funding for this work was provided by the Hari Center here at the U of A, and has since the beginning of time, at least my time here at the U of A, has been super supportive of our work and uplifting Indigenous voices and knowledge. I hope I didn't go over time too much, but I'll stop there and take your questions. I really appreciate everyone's time and attention and all of your energy today. Thank you so much.
Thank you, Dr. DeVore, and outstanding presentation. It's really wonderful to hear about all your research, how you have connected that to back to your own indigenous worldviews and working with our people, and then mentoring the next generation of scientists and engineers. So thank you so much. We appreciate you sharing that today. At this time, we'd like to open it up for Q&A. So if you have a question, please go ahead and raise your hand and you can verbally ask the question. If you would like to also put it in the chat, I can also read it for you. Great. Annalise Guthrie, could you go ahead and ask your question? Thank you.
Hi, Dr. DeVore. I really loved your talk. This has always been really interesting to me. I'm a soil scientist myself. I like call myself more of an ecosystem ecologist. So this is always, I came to your talk because I was like so interested in your work already, and thank you for your presentation. I was just curious. I don't have like much experience in like studying remediation at all, but I was just curious if you have thought about at all how maybe monsoon season or climate, specifically precipitation, might influence the efficiency of your of this fungi to remediate these metals at all. If that sort of dilution of the soil water might change how the dilution of the arsenic or the heavy metals might change how the effective the fungi are able to absorb it, I guess.
Yeah, that's a really great question. And I think it's important to think about what the growth requirements are of fungi. So you have the majority of what we're working with are filamentous fungi, and they require like 60 to 70% soil moisture saturation. So if you're going to do it in situ, that would be an incredible challenge here in the desert Southwest, right? So growing in a laboratory setting and growing for like scale is something we're navigating and working towards because that is the case. Secondly, Ryan Cotter, who's leading work with Podaxis, who we were joking the other day is like harsh loving mushroom. So compared to mostly what I've been talking about, filamentous fungi, Podaxis is a fruiting mushroom, an actual mushroom, and grows in like rocks with no water, with like super harsh heat and cold conditions. And so at this time, we're piloting with her and we're thinking about growth requirements and what it would take for Podaxis to thrive. That's a first step. And in the long term, you bring up really great points, right? About monsoon, about even just solubilizing metals in situ and the capacity for these materials to biosorb under those circumstances. We're not quite there yet, but that's a really great question. And
We have another question by Caitlin Haskey.
Hi, Dr. DeVore.
I,
Sorry, I Nice to see you. This is a great presentation. It's very interesting. I always say I took a mycology class my final semester at NAU as an undergrad, and I always say that if I had taken one sooner, I probably would have just I think fungi are so cool. Uh, so that being said, I do have a couple of questions. I was kind of a little bit late, so forgive me if I missed this. But I was wondering if you've identified what transporters are the fungi use that are part of the bioremediation process. And then I was also wondering how long have you noticed it takes the fungi to remediate some of those metals? And then as a follow up to the last question somebody posed, I know that plants are heavily controlled by their environment. And so I think if you're pulling this fungi from, I'm just kind of wondering where you got the samples from, because if you're pulling them from like home, the way that those fungi are conditioned, how they respond to the environment is, it really dictates how they function. And so I was just kind of wondering where you guys get your fungi, and then how is that kind of mimicking the natural landscape in the Southwest? Thank you again.
Yeah. So your first question is a Savia question, who's leading the transcriptomics and metabolomics, but the first enzyme coming to mind is ARS one for uptake into the vacuole, which then can be volatilized. That's the one coming to mind. And then your second question on where we got our fungi, we isolated these from the landscape from several areas across that longitudinal map that I showed. So metal tolerant filamentous fungi that co-occur with plants that are also tolerant of metals. And so Savia is leading that work. And then you're right, you make a really great point on the very specialized environments, whether that's soil chemistry, soil water characteristics, nutrients, different types of ecosystems have different types of fungi and plants that are associated with those fungi. And so we're not drawing directly at this point in time from areas here in Arizona or in the Southwest. We're using fungi that are very well characterized, not just in the work that I've done, but in the literature broadly on who is metal tolerant, and learning from those mechanisms to then, once I get relationships on the ground here in the state of Arizona, we have a toolkit and resources ready to go for folks that would like to collaborate. And I haven't done so thus far because I've been spending these 2 years getting to know people in the community and becoming like a relative and a community member, and not just like inserting myself and the tools into areas where I'm not asked. So I hope that answers your question.
Okay, thank you, Caitlin, for asking that question. We do have one question in the chat, and then after that, I'll go to Nico. So the question in the chat is, are these fungal species pathogenic to humans or potentially to wildlife?
Some endophytes are pathogenic to wildlife, but I think animals know know the landscape better than we do, and so they don't eat the plants with these specific fungi. And so maybe I didn't clarify well enough in the presentation, but we would not be taking fungi and introducing them to a landscape, because there's some potential impacts there, as you can imagine. What we're thinking of doing is either in a settling pond, either in columns, either in batch reactors, or like floating microcosms in areas impacted by mining, or growing mushrooms on the mine waste. These are what we're thinking in the long term. But in order to get there, you have to figure out, of these metal tolerant known species, who is best at adsorption.
Thank you, Dominic Rodriguez, for asking that question. He's the director of the Indian Cooperative Extension Program at the U of A. So thank you, Dominic, for joining us. And we look forward to more of the agents joining us as well. We do have a question from Nico Sanchez. So, Nico, if you could go ahead and unmute yourself and ask, that'd be
Yeah.
great.
Thank you so much for the presentation. Um, I studied ecology and evolutionary biology during my undergrad, and one thing that I really appreciated is from this presentation is kind of like the parallels between like the work that you're doing as well as like the work that you're doing within community, both here at the university and beyond. Um, But as we know, like nothing really kind of happens in isolation, especially microbially. Um, can you talk about like how the impacts from other types of microbes might like affect this work and have some kind of impact on these endophytes?
Yeah, that's a really great point, Nico. Thank you so much for joining and thank you for the stickers. Um, I like to draw on my friend Luis Barrios, who's a microbial ecologist, and he's articulated what's known as like tripartite systems of not just fungi, but bacteria and plants. You're not going to go into any ecosystem anywhere and not find these three together. So if the figure here didn't articulate, let me just go this one. So I work in layers. I build on complexity. So there's part of it that's like water only, soil and water, soil, water, and plants, soil, water, plants, bacteria, fungi, and then eventually all of them together into these very complex understandings. Whereas if we were to do the backwards approach, right, of all of these together, it would be really hard to tease out and understand the different layers that add to the complexity. So you're absolutely right, Nico, on what role, for example, bacteria play on interactions with fungi. And that's something that we are thinking about in this laboratory on designing experiments with bacteria. And then Savia is leading the work with her phytoremediation project in the long term with plants, bacteria, and fungi together. It gets very complex very, very quickly, very quickly. And so to not overwhelm the students and to not overwhelm the research questions, we're working in layers.
Yes, that's a great way to think about it. I like your figure there. So I want to just thank you, Dr. DeVore, for sharing your insights and your experiences with us today. We appreciate your time and your knowledge that you shared. And also a big thank you to everyone who's joined us here and our co-sponsors, the Indigenous Resilience Center, the U of A Sloan Indigenous Graduate Partnership, Water Resources, the Howry Program and Social Environmental Justice, the Office of Societal Impact, for all making this seminar possible. We invite you to join us for a session after this, which is focused on student mentorship. So any students on the line, feel free to stay on for a one-on-one discussion with Dr. DeVore. And look out for us in 2026 for our spring Native Voices in STEM seminar series. And we'll be releasing that pretty soon. We really like having everyone here, having the Q&A, and just continue to look out for our next announcement. So thank you so much, and we look forward to seeing you next time.
Thanks, everyone. Appreciate you.